And please note that this is the part 1 of the lecture. Before starting this lecture, I just want to give you more information that the images, text and other materials used in the presentations of education purposes only. We respect the intellectual property rights of the original creators and owners. If you are the owner and have any concerns or issues, please contact us at sathee@itk.ac.in and we shall remove the content immediately. Please note that the parts of the content may have been generated using generative AI, which is experimental. So what are the objectives of today's session? So we will discuss the Mendel's law of inheritance. So Mendel, he is known as the father of the genetics, and he gave laws of inheritance. Okay, so we will understand what is inheritance and what is variation, and then we will understand the Mendel's law of inheritance. Okay, so we'll talk about the law of dominance, law of segregation. Okay, and there is one more law, law of independent assortment. We'll also talk about that, but in the next session. We'll talk about the inheritance of one gene. So within this, we'll talk about the monohybrid cross. Okay, we will understand the inheritance patterns of single gene. So crosses which involves only single gene or single trait, they are known as monohybrid cross. In monohybrid cross, we study the inheritance of one gene. So we'll study the inheritance patterns of one gene through monohybrid crosses, and we will predict the genotypes and the phenotypes of the offerings using the Punnett square. Then we'll differentiate between dominant and recessive alleles. So we will understand what is dominant allele, what is recessive alleles, and what are alleles. And we will also discuss how these alleles, they affect the phenotypic expression in the homozygous and the heterozygous conditions. Okay. So in this session, we'll be talking about Mendel's law of inheritance. Okay, which is section 4.1. We'll also talk about the inheritance of one gene, which is section 4.2. Then we'll discuss some questions which are related to this topic, and we will, in the end, we will be discussing some keynotes which will help you in the NEET exam as well. And these keynotes will help to memorize also. Okay, to help you memorize the concepts related to these topics. Now let's begin with this session. So let me first introduce you to this chapter. Now, what is the mystery of inheritance? What is inheritance and what is the mystery behind it? So you can see elephant only give birth to an elephant, not any other organisms. Okay, so the elephant, okay, the offerings of an elephant resembles the elephant in morphology, in size, okay, when it grows up. And a mango seed only grows into a mango tree, not into a strawberry or any other tree. So why is this happening? Okay, we'll discuss this. Now, offerings, let's talk about offering resemblance and the variation. So offerings are not identical to their parents. So sometimes what happens, okay, especially during in the sexual reproduction, the offerings, they are not identical to their parents. Because what happens in sexual reproduction? Male and female, it means two parents are involved. Biparental conditions is there. Male and the female, they produce gametes, male and the female gametes. Gametes fuse with each other. Means the genome of the parents, they mix together. So this leads to variation. And also when the gametes are formed by meiosis, then recombination and crossing over occurs in meiosis. This is also a source of variation. So offerings, especially in the sexual reproduction, they are not identical to their parents. Okay. So this is due to genetic recombination and random inheritance of the genes. So during sexual reproduction, what happens? Gamete formation occurs. Gamete formation occurs by meiosis. During meiosis, recombination and crossing over takes place in the Pachytene stage. Clear? So this is one source of variation. And then the, you can say, the genetic makeup, the genome, okay, the genes of the mother and the father, they are combined together in the zygote. So in the zygote, one set of chromosomes comes from the father, another set of chromosome comes from the mother. So there is mixing of the genes. This is also a source of variation. Now, siblings can look alike. Okay, the siblings, the children's, they can look alike as well because they share genes from the same parent. Clear? So the sisters, the brother, okay, they sometimes look alike. So why do they look alike? Okay, sometimes they share a lot of similarities. So if you see them, you say, okay, they are related. You can tell them by seeing them. So siblings, they look alike. Why? Because they share genes from the same parents. Although there are a lot of differences, but they share some genes. That's why they resemble each other. Now, siblings can look different also because each sibling receives a unique combination of parental genes. Okay, so siblings can look alike, but they are different also. Okay, why they are different? Because they receive a unique combination of parental genes. Now let's understand some key concepts in the genetics. Now, first of all, what is genetics? Genetic deals with the inheritance and variation of traits from parents to the offsprings. So this is a very simple definition of genetics. Okay. Here two terms are written, inheritance and variation of the traits. So these inheritance and variation, genetics deals with these two. Okay. Now let's define inheritance. What is inheritance? So inheritance is the process of passing traits from parent to the offsprings. And it is the basis of the heredity. So what is happening in inheritance? The traits, they are passed on from the parents to the offsprings. The genes, they are passed on from the parents to the offsprings. This is known as inheritance. Okay. Now, inheritance is the basis of the heredity. Okay. What is variation now? A degree to which progeny differs from their parents. This is known as variation. Or simply you can say progeny differs from their parents. Clear? Now the progeny is different from their parents because they inherit unique combination of the genes. And there is recombination also. Clear? And there is random inheritance of the genes. So the progeny is different from the parents. So they show variations from their parents. So the degree to which the progeny differs from their parent, this is known as variation. Means how much they are different from their parents. This is known as variation. Okay, variation is very important. And sexual reproduction is also very important. So what is the significance of sexual reproduction? It leads to variation, and variation leads to evolution, because this variation, they keep on accumulating over generations and it leads to evolution. Now, early understanding of the variation, let's discuss this. Okay. Now let's talk about the period 8000 to 1000 BC. Now, humans understood that the cause of variation was hidden in the sexual reproduction. So our ancestors, the early humans, so they had the knowledge that these variations in the progeny, they occur due to sexual reproduction. So they had this knowledge, but they didn't know the mechanism how these variations occur. Okay. So by just observing Okay, they used to just observe, okay, the childrens, they are different from their parents. They don't look like their parents. Although they have some similarities, but they are not same. Okay, there are some differences. Clear? Now what they did selective breeding. So humans exploited naturally occurring variations in the wild plants and animals to selectively breed and select for desirable traits. Okay, so what early human or our ancestors did then? They exploited the naturally occurring variations in the wild plants as well as the animals, and they start selectively breeding. So what happened? So now they knew, okay, this plant has this characteristic, this animal have this sort of characteristic. So they want these characteristics in the progeny also. So they chose that plants which have that desirable characteristics, and they you can say intercross them. So this is known as selective breeding. So here it is written select for the desirable traits. So what is important for selective breeding? You have to choose the parents first. These parents should have the desirable traits or the desirable characters that you want in the progeny. Then you cross these parents, and there are high chances that in the progeny, these characters will be acquired from the parents. Okay. So human was doing all of these activities for its own benefit, because they want plants with high yield, they want plants with high vigor, they want plants which should have stress tolerance, they want animals, okay, such as cows, okay, which can produce more milk. So he was doing artificial selection and domestication. So through artificial selection and domestication, ancestral wild cows were bred into well-known Indian breeds such as Sahiwal cows in the Punjab. So through selective breeding, okay, a variety of this cow known as Sahiwal cow, these cow have been developed in the Punjab. So ancestral wild cows, they were bred into a well-known Indian breed such as Sahiwal cows in the Punjab. So this is Sahiwal cows in the Punjab. They, okay, so these breeds, they have been made through artificial selection and domestication. Now there was limited scientific understanding in the earlier days. So our ancestors, as we have discussed, they knew that these variations, they occur due to sexual reproduction. Okay, they understood inheritance of characters and variation, but they lacked a scientific understanding of the underlying mechanism. So they didn't knew the mechanism of this, how this variation arise. So they lacked the scientific understanding. Okay, so they didn't knew about the DNA, they didn't knew about the genes. Now we know that genes are inherited, and genes, they are the segment of the DNA. So basically DNA is being inherited from the parents to the progeny, and DNA is carries all the information. Okay, the genes, they code for a particular trait. Okay, clear? So genes are passed on from the parents to the offerings. This is known as inheritance. And genes control a particular trait. So inheritance of trait from parents to the progeny, this is also known as inheritance. Okay, so this inheritance was studied by Mendel. That's why he's known as the father of genetics. Now the dawn of inheritance understanding. Okay, now comes the role of Mendel. We'll study about the Mendel's law of inheritance now. Okay, so our ancestors, they didn't know about the how the inheritance, okay, is occurring, how the variations are arising. So a significant process in understanding the inheritance occurred during the mid 19th century. Okay. Now let's talk about the Mendel's contributions. So Mendel's name was Gregor Johann Mendel. This was his full name. He's known as the father of the genetics because he gave the laws of inheritance. Now, Gregor Mendel conducted extensive hybridization experiments. Okay. So he was working on the pea plant. So he was doing this hybridization experiment in the pea plant. So his experiment involved garden pea, means pea plant, and his experiment, he conducted experiments for 7 years from 1856 to 1863. So you can see how much work was done by Mendel. And finally, he formulated the laws of inheritance in the living organisms. So this means that how the traits are inherited from the parents to the offsprings. This was, you can say, deciphered by Mendel. Now, Gregor Mendel's contribution. Now, Mendel's work pioneered the use of statistical analysis and mathematical logic in biology. So, Mendel's work was quite significant. Okay, it is very significant actually. So, what did Mendel do? So, Mendel was the first person to use the statistical analysis and mathematical logic in the biology. So, he was one of the first person. So, he pioneered the use of statistical analysis and mathematical logic in biology. And this was not accepted by the other biologists because they were against the use of the statistical analysis and mathematical logic. So a lot of the scientists at that time, they rejected Mendel's work. Okay, but this was actually good. Statistical analysis is always good for the results. Now, Mendel's experiment utilized a large sampling size, significantly enhancing the reliability of his data. So always, whenever an experiment is done, the sample size should be large. Okay, you should analyze more number of samples because if you analyze more number of samples, there will be a lot of variations. And okay, so this significantly enhances the reliability of the data if you are experimenting on large population size. Now, confirmation of inferences or experiment of successive generation of the test plant. So what Mendel did? So Mendel carried out his experiments on successive generations. Clear? So he confirmed the result in the successive generations also. Now, Mendel's result indicated general rules of inheritance, not substantial ideas. So you can see, because of all of these points that we have discussed, Mendel's laws of inheritance, they were based on the experimental proof. Mendel gave experimental proof for his results, which lead to general's rule of inheritance. And it was not just substantial ideas. His results were based on experimental data, okay? And he carried out his experimental successive generations. Now, Mendel's fundamental genetic research and methods. How did Mendel carried out the experiments? Mendel created basic rules for the inheritance patterns. Now, the later scientists built upon and expanded Mendel's rule. So initially, Mendel gave the basic rules of inheritance. Then the other scientists, they carried on his work and expanded the Mendel's laws of inheritance. What Mendel did? Mendel carried out artificial pollination in his experiments with the pea plant. Okay. Mendel's experiment involved multiple pure breeding pea plant varieties. So this is very important point here. First of all, Mendel carried out artificial pollination. The question can come from here. What type of experiment did Mendel carried out? Now, Mendel used pure breeding pea plant varieties. Pure breeding pea plant, or you can also write true breeding pea plant varieties. Okay, what do you mean by pure breeding pea plant varieties? So this pure breeding pea plant varieties or pure breeding varieties, they are created by repeated selfing, by repeated self-pollination. Okay. So because of repeated self-pollination, these pure breeding varieties, they are homozygous for a particular trait. Okay. Means their genotype will be capital T, capital T. If we talk about the height as a trait, or small t, small t. So they are homozygous for a particular trait because they are created by self-pollination and they show stable trait inheritance. Okay. Means in the next generation also, the traits will be inherited. Okay. This is the meaning of stable trait inheritance. The traits will not be lost in the offering. Sometimes the traits, they are lost in the offerings due to sexual reproduction or due to segregation of the genes, due to independent assortment of the genes. The traits are lost in the offerings. But if the varieties are pure breeding, then the traits will be maintained in the next generation also. So a pure breeding line is one that have undergone continuous self-pollination, means selfing, and shows stable trait inheritance and expression for several generations. Means the trait will be maintained from one generation to next generation. Now, as we have discussed, Mendel carried out artificial pollination and cross pollination. Okay. Yes. Now we'll discuss how Mendel carried out these cross pollination or artificial pollination. Okay, I'll tell you why it's called cross pollination and artificial pollination. So what is cross pollination? So when the pollens, they are transferred from the anther to the stigma of another flower. This is known as cross pollination. What is self pollination? When the pollens from the anther of one flower, they are transferred to the stigma of the same flower, that is known as self pollination. If you transfer the pollen to the stigma of another flower, that is known as cross pollination. So Mendel carried out cross pollination. Okay. Now, why it was called artificial pollination? We'll discuss this also. Now, first discuss Mendel's choice of traits. So Mendel chose 7 pair of contrasting characters. So Mendel investigated characters in garden pea plant. So he was working on garden pea plant. Now he focused on, or you can say he was studying on 2 opposite traits per character. Means each character, each trait had 2 opposing phenotype. For example, if you take height as a character, the plants were tall or they were dwarfed. If you take color of the seeds were yellow or they were green. So you can see here, these are 2 contrasting traits of a particular character. These are 2 phenotypes, yellow and green, tall and dwarf. These are 2 phenotypes. These are 2 phenotypes of a single trait or a single character. So Mendel, he was working on, or he focused on 2 opposing phenotype per character. So Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. So what does this mean? So Mendel chose 7 pair of contrasting character. Means he chose 14 true breeding plant varieties. Okay, so when he crossed, Mendel crossed the pea plant. So pea plant was differing in. So he crossed 2 pea plant. One of the plant had yellow seeds, another plant had green seeds. Then he did experiment on another pea plant. This pea plant had tall height and the other pea plant has short height. So he crossed these 2. So there were 7 pair of contrasting characters. So in total there will be 14 plants. And he was working on true breeding pea plant varieties. so that stable trait inheritance was observed. So he selected 14 true breeding pea plant varieties. Why 14? Because he was working on 7 characters and the 7 character had 2 phenotypes. So 72 will be 14. Now 7 pair of contrasting characters chosen by Mendel in the pea plant. Let's have a look at this figure 4.1. So you can see here, these were the characters. Seed shape. Seed shape can be round or wrinkled. Seed color can be yellow and green. Flower color can be violet and white. Pod shape can be full or constricted. Pod color can be green or yellow. Flower position can be axial or terminal. Stem height means the height of the plant can be tall or dwarf. So you should remember these 7 pair of contrasting characters. So there were 7 pair of contrasting characters. So there will be in total 14 true breeding pea plant varieties. So what did Mendel did? Mendel crossed a plant with round seed shape with a plant with wrinkled seed shape, and then separately Mendel crossed a plant with yellow seed color with a plant which have green seed color. Then Mendel crossed a plant in which the flower color was violet. He crossed this plant with another pea plant which has white color flowers. Okay, so he chose 14 true breeding plants. You should remember these 7 pair of contrasting characters also. So again, you can see in this table, these are the 7 pair of contrasting characters chosen by Mendel. What were the characters? Stem height, flower color, flower position, pod shape, pod color, seed shape, and seed color. And these were the contrasting traits. So you should remember this table. Now let's talk about hybridization experiment conducted by Mendel. So first of all, Mendel chose the parental generation, which is written as P. So let's take an example. Suppose Mendel crossed a pure breeding tall plant with a pure breeding dwarf plant. So these are pure breeding, means they are homozygous for a particular character. So the tall plant will have genotype capital T. Pure breeding dwarf plant will have small t. Okay, both are pure breeding. They have been created by repeated selfing. Now what will be the gametes produced here? So this homozygous tall plant, this will produce only T gametes. Similarly, the plant with genotype small t means homozygous recessive plant. This homozygous, this is dwarfed. This will produce only small t gametes. So what will happen after crossing these two parents? In the F1, what we will get? So we can make a Punnett square. So here you will write the gametes on one side of one parent. In the other side, you will write the gametes of other parents. And then you can make different combinations in the Punnett square. So here you can see what genotype you are getting. You are getting capital T and small t. So 100% of the F1 generation is capital T, small t. This is the heterozygous conditions. Heterozygous condition is where dissimilar alleles are present. In homozygous, similar alleles are present. So F1 generation, also known as filial 1 generation, this is the first generation. So all plants will be tall here and the genotype will be capital T and small t. So what did Mendel observe? All the plants were tall. Okay. And you can see in F1 only one of the character is observed. Means all the plants are tall. No plants were dwarf. Okay. So that's why the F1 generation mirrored one of the parental plants only. Only one phenotype was observed. Only dominant phenotype is observed in the F1 generation. No dwarf plant means no recessive trait was observed in the F1 generation. And 100% of the plants were heterozygous tall. Clear? Now, Mendel did artificial cross-pollination. So what did Mendel do? Mendel selected the plants. As we have discussed, Mendel selected a tall, pure breeding tall plant. He crossed it with pure breeding dwarf plant. And then Mendel emasculated the female plant. He emasculated the plant to make it female plant, because the pea plant, the flowers of the pea plant, they are bisexual, means they have both the reproductive organs in the same flower. So emasculation means removal of anther was done to make the plant female and also to prevent the self-pollination. Because if both the reproductive structures are present in the same plant, then there are chances of self-pollination. So to prevent self-pollination, emasculation of the flowers was done. Okay, Mendel wanted cross-pollination. Now then he applied the pollens from the male flower to the stigma of the female flower, which he made by emasculation, and then bagged it so as to avoid contamination with the pollens from any other plant. So that's why it is known as artificial cross-pollination. So here are the steps in making a cross. So Mendel, what he did. So this is the pea plant here. This is the flower of the pea plant. So you can see this pea plant consists of stamens as well as the, you can see here, this is the stigma, which is part of the carpel. So carpel consists of ovary, style and stigma. So this is bisexual flowers. Both the reproductive organs are present here. So what Mendel did, he removed the anthers. And this is known as emasculation. Emasculation was done to prevent the self-pollination. And then after emasculation, after removal of anthers, this flower, this becomes the female flower. And then this is the male flower here. What Mendel did, he artificially pollinated the female flower now. So that's why it is artificial cross-pollination. So pollens from the male plant, this is the symbol denoting the male, this is symbol denoting the female. Now pollens from the male flower, these were dusted on the stigma of the female flower. This is how the artificial cross-pollination was done. And now after pollination, the F1 generation was observed. So after pollination, we will get the first generation, which is known as the F1 generation. In F1 generation, all the, you can say, plants were tall. Now selfing of F1 generation was done to obtain the F2 generation. F2 generation is phylyl 2 or second generation. Okay. So selfing of F1 generation means, or you can also call F1 generation as F1 hybrid, means this genotype will be crossed. Capital T, small t will be crossed by capital T, small t. Now you can again make a Punnett square. So you will write the gametes of the one parent on one side and gametes of the other parents on other side. So here what gametes will be produced by each parent? Capital T, small t. Now by making a Punnett square, you can make different combinations of the genotype which will be produced. Okay, now observe this table. Okay, here capital T will be formed here. Capital T, small t, capital small t, and small t is also formed here. So you can see the recessive character, recessive trait is appearing in F2 generation. This was not appearing in the F1 generation, but recessive trait is reappearing in the F2 generation. So in the F2 generation, what is happening? 1/4 means 25% had the genotype capital T, and its phenotype will be tall. Then 50% or 2/4 will have a genotype capital T, small t, and these plants will also be tall. Then 1/4 or 25% will have the genotype small t, and these plants will be dwarfed. So recessive condition, this is always observed in the homozygous state. Okay. Now this tall or the dominant trait, this is observed in the heterozygous and the homozygous condition as well. Now this demonstrated the reappearance of the recessive trait in the F2 generation. So in the F2 generation, the recessive trait, it reappeared. So here you can see this is the cross. This is the diagrammatic representation of the monohybrid cross. Monohybrid cross means a cross involving only one trait or one gene. Parents were tall and dwarfed. These were pure breeding lines. In F1 generation, all the plants were tall. F1 generation was selfed. So there was selfing of the F1 generation. And in the F2 generation, you can see tall plants as well as dwarf plant was observed. So what is the genotypic ratio here? Genotypic ratio will be 1:2:1, where 25% means 1/4 are homozygous dominant. 2/4 means 50% are heterozygous, capital T, small t. So this 2 is capital T, small t, 1 is capital T, capital T. And then 1/4, 25% are homozygous recessive, small t, small t. So this again 1, this is small t, small t. So genotypic ratio in a monohybrid cross, this is 1:2:1. So question can come from here. Then what will be the phenotypic ratio in the monohybrid cross? Yes, it will be 3:1. Phenotypic ratio is 3:1. Okay, 3 tall and 1 dwarf. So 3/4, 75% show the dominant trait, means they are tall, and this includes both the genotype, capital T and capital T small t. So we'll discuss this later. This is because of the dominance of the genes. Capital T is dominant over small t, and 1/4 means 25% show the recessive trait, and this includes small t, small t. This recessive trait is only observed in homozygous condition. Now, what are the important points of this cross that Mendel did? So here you can see the dominant allele capital T masks the recessive allele, which is small t in the heterozygous individuals. Okay. Then the trait only appears when both alleles are recessive. The recessive trait will only appear when the alleles are homozygous. Okay. Now multiple genotype capital T and small t can produce the same phenotype, which is tall. So you can see these are two different genotypes, capital T, capital T, capital small t. They are all producing the same phenotype. Now the contrasting trait, they did not blend in either the F1 or F2 generation. So you can see there was no blending of the trait. What do you mean by blending of the trait? So blending of the trait means there was no intermediate phenotype. The plants were tall or they were dwarfed. So there was no plant of intermediate height. So means there was no blending of characters here in the F1 or in the F2 generation as well. So this is an example of complete dominance. Now what was Mendel's conclusion here? So based on observation, Mendel proposed that something was being stably passed down from parents to the offerings through the gametes. So what did Mendel said? Okay, something is being stably passed from the parent to the offerings, but he didn't say what is being passed on. He only said something is being stably passed from parent to the offerings. Okay, this something now we know this is known as the genes. So now we know genes are inherited. Genes are passed on from the parent to the offerings, and this is known as inheritance. Now let's talk about genes as the unit of inheritance. So genes, they are the fundamental unit of inheritance. And let's talk about genes and the trait. So genes, they control a particular trait. So genes, they carry the instruction for the expression of a specific trait within an organism. So genes, okay, they control a particular character, they control a particular trait. And gene is a segment of DNA. Now what are alleles? Alleles, they control the contrasting traits. So what is the simple definition of allele? Different form of the gene. This is known as allele. So genes that determine contrasting form of a trait, these are known as alleles. Or simply you can say alleles are different form of the genes. So how alleles are produced? So these two alleles, they are different form of a same gene. And these two alleles, they differ slightly in their DNA sequence. And this difference may arise due to mutation. Clear? Now, these alleles are slightly different versions of the same gene. And how these two alleles, they arise? Maybe due to mutation. These two alleles are very similar to each other. Okay. There is only slight difference in their DNA sequence. Now, Mendel's finding on pea plant height. So, in true breeding tall plants, allelic pair of height is identified as capital T, capital T. So, this is homozygous dominant. Okay, this was observed in the F2 generation. Now, small t means homozygous recessive was also observed. This was also observed in the F2 generation. So when the two alleles are identical to each other, these are known as homozygous. Homozygous can be homozygous dominant or homozygous recessive. Now heterozygous was also observed. Now a plant with a genotype capital T small t, this is heterozygous, means having two different alleles. Now in this case, what will be the phenotype in capital T small t? Here the dominant trait will be expressed. So tallness is the dominant trait. So that's why capital T small t will show a, you can say all the plants will be tall because tallness is a dominant trait. So the plant having capital T small t, they will be tall. Now let's understand what is genotype and phenotype. So capital T and small t, these are the genotype. Means the genetic makeup of any individual. This genetic makeup is known as genotype. Okay, and the observable trait, this is known as the phenotype. So tall and dwarf, they are the phenotype. Okay, the trait which you can observe. So you can observe the plants are tall and they are dwarfed. This is known as the phenotype. Now what is monohybrid cross? So simply a monohybrid cross is a genetic cross that studies the inheritance of single trait or a single gene, you can say. Okay, this single trait is controlled by a single gene and this gene have two different alleles. Like for height, capital T and small t. Capital T for tallness, small t is for dwarfness. So this is known as the monohybrid cross. Simply you can see a cross between two individuals which differ in only one trait or one gene. Example, we take the character plant height. So plant can be tall or dwarfed, or you can take the flower color. Flower can be purple or white in color. So here when the cross is made, the plant, they differ only in one trait, which are controlled by only one gene. Let's briefly talk about law of segregation also. Now the reappearance of the recessive parental trait. Recessive trait was dwarfed. This trait appeared in the F2 generation. This demonstrates that the alleles separate or they segregate during the meiosis. So segregation of gametes occur during the meiosis. Remember, this question can come from here. Now each gamete receives only one allele from the parental pair. Okay, so the gametes will receive only one allele. So in the case of heterozygous capital T small t, half of the gamete will contain capital T, half of the gamete will contain small t. Okay, now allele similarity and dissimilarity. Alleles can be similar in homozygous and alleles can be dissimilar in heterozygous. We were talking about segregation of the allele. So segregation of allele is a random process. There is a 50% chance of gamete containing either allele. Okay, like in the case of capital T and small t, 50% of the gamete can have capital T, 50% of the gamete can have small t. So this can be verified experimentally. Okay. So in the genotype capital T, all the gametes will have allele capital T. In the case of genotype small t, the gametes will have only small t. Okay, in the case of heterozygous, it is different. Now, what happens when we cross these two plants? Plant having capital T and plant having small t and small t. During fertilization, the alleles unite to form in the zygote. So in the zygote, half of the chromosome come from the mother, half of the chromosome come from the father. Or you can also say in the zygote, one allele will be acquired from father, one allele will be acquired from the mother. If we talk in the sense of allele, so in the zygote, what will happen? Capital T and small t will be formed. So example, capital T is coming from the pollen and this combines with the small t coming from the egg. Okay, so zygote will have capital T and small t. Okay, so zygote will be heterozygous. It will have capital T and small t. So this will be known as a hybrid, which will be heterozygous. It will have capital T and small t. Okay, now let's understand the inheritance with the help of the Punnett square. So first of all, what is Punnett square used for? So Punnett square gives you information regarding the production of gametes by the parents. Okay, then it will tell you about the genotype of the zygotes or the hybrid which is formed. Okay, and then by looking at the genotype, you can tell the phenotype also. Okay, then the F1 generation and F2 generation, the genotype of F1 and F2 can be understood by the Punnett square. So here you can see in this diagram, a tall parent is crossed by a dwarf parent. You will write the gametes here. Gametes of one parent on one side, gametes of another parent on the other side. Then you will get the F1 generation. Then F1 generation is self. You will get the F2 generation. So this is the Punnett square. In the Punnett square, you can see the gametes here. So it gives you information regarding the gametes produced by the parents, the formation of the zygote means the hybrids, and the F1 and the F2 generation as well. And here you can see the phenotypic ratio, genotypic ratio is written here. In the monohybrid core, genotypic ratio is 1:2:1, phenotypic ratio is 3:1. Okay, so gamete production by the parents, zygote formation, F1 and F2 plant development, these can be understood using the Punnett square. So these can be visualized using a Punnett square. This was developed by a British genetist, Reginald C. Punnett. That's why the name Punnett square. What is the purpose of Punnett square? So it's first of all, it is a graphical tool. It calculates the probability of all possible genotypes of the offerings in a genetic cross. So remember this. This is the purpose of the Punnett square. Okay, all the possible genotypes of the offerings in a genetic cross can be calculated using a Punnett square. Okay, so here gametes are placed. The possible gametes are listed along the top row and left column of the Punnett square. Okay, then you get the output. All possible offering genotype are represented in the squares within the grid. So the Punnett square is like a grid, and within this grid, all the possible genotypes are listed within this squares within the grid. So this creates a square output form. Okay, F1 generation is produced from the parents, and the selfing of the F1 generation is done. Okay, clear? Now you can see this symbol is for the male and this symbol is for the female. So this denotes the pollen and the eggs. Pollens are the, you can say, they form the male gamete, and eggs, they are the female gametes. So they can be represented using these symbols. Clear? And then you can obviously show the gamete production. Okay, you can show the selfing of the F1. You will get the F2. You can tell the genotypes of the F2. Okay, then the hybrids which are produced. So these hybrids which are produced in the F2 generation, which we get after the selfing of the F1. In the F2 generation, what all genotype will be produced? Capital T, capital T, capital small t, and small t, and small t. Okay. So this we have already discussed. 1/4 will be capital T, 1/2 will be capital T, small t, and 1/4 will be small t and small t. Means 1:2:1 ratio. Okay. In the F1 generation, we have already seen that the phenotype was tall. So this was because of dominance of one gene. In the F2 generation, 3:1 phenotypic ratio is observed. Okay, clear? This is all we have already discussed. So plants with genotype capital T and capital T, small t, they have the same external appearance, means they have the same phenotype. Clear? So phenotypic ratio is 3:1. This we have discussed. And the genotypic ratio will be 1:2:1. Okay. Now you can see here, this ratio 1 is to 2 is to 1. This can be expressed using a binomial expression, which is ax plus by whole square, which is nothing but a plus b whole square. Okay, so this equation represents the possible combination of the gametes. Okay, so gametes carry either the capital T or small t allele, and each with the equal frequency of half. So if this is the genotype, capital T, small t, half of the gamete will carry capital T, half of the gamete will carry small t. That's why it is written, they have a equal frequency of half. Okay, so you can put this half here in this equation, half capital T plus half small t whole square. And this can be expanded as follows, half t plus half small t into half capital T and half small t. So this is the formula for this a plus b whole square. And then if you calculate, you will get 1 by 4th capital T, half capital T small t, and 1 by 4th small t. Okay, clear? So this is the binomial expression of the combination of the gametes. Now, then again, as we have discussed, Mendel carried out on his experiment on the successive generations. So then Mendel self-pollinated F2 plants also. And he observed that dwarfed F2 plant produce only dwarf plants in the subsequent F3 and F4 generation. So what does this mean? If you are getting the dwarf plants in the F3 and F4 also, means this dwarf plant will be homozygous. Okay? Clear? If it is homozygous, then only in the next generation, the recessive trait will be produced. Clear? So you can see Mendel concluded that dwarf plant has a homozygous, means small t genotype, because in the F3 and F4 generation, dwarfed trait was observed. Okay, now what was the challenge to the dominant traits? So let's understand. So we'll talk about test cross here. Now you can see in the F1 generation, heterozygous condition was observed, capital T and small t, and the phenotype was tall. Okay. Now how to determine whether the genotype of this tall individual, that will be capital T or small t. So it is impossible to determine the genotype of an organism solely based on its phenotype when the trait is dominant. So you cannot tell. If you see the plant, pea plant is tall. So by looking at the plant, you cannot tell that its genotype will be capital T, or it will be capital T small t, means it will be homozygous dominant, or whether it is heterozygous. You cannot tell by just looking at the plant if it is tall. Okay. So observing a tall plant does not definitely reveal whether its genotype is capital T or capital T small t. So for this, you have to do a test cross. So what is test cross now? So crossing an organism with a dominant phenotype. Dominant phenotype means, suppose let's take the example of tall plant. Tallness is a dominant phenotype. So plant are tall, but its genotype is unknown. So you cross the organism or individual with dominant phenotype with a recessive parent, which is homozygous recessive. And then by this cross, you can tell whether the genotype of this dominant parent, this is homozygous dominant or heterozygous. So you can see here, let's understand this using this image. So you can see here, homozygous recessive is small w, homozygous recessive here again small w. So what is being done here? You want to know the genotype of the dominant phenotype here. This, you want to know the genotype of this individual, where the question mark is written here. Okay. So this individual has a dominant phenotype. So this individual can have two genotypes, capital W, which is homozygous dominant, or it can have the genotype capital W and small w. Now what is done? The dominant phenotype, this individual is crossed with homozygous recessive. Now let's take a look at this cross on the left hand side. Now if the genotype is capital W, then you will get all the flowers will be violet here. Okay, you can make a Punnett square. You will write the gametes here. On one side, you can write the gametes of homozygous recessive on the other side. The gamete produced by capital W will be W only. Gametes produced by homozygous recessive, this will be W only. So we will get all the flowers will be violet only here. Okay, if all the flowers are violet, then the genotype will be capital W means it will be homozygous dominant. Now, in the other case, on the right hand side, you can see if the genotype is capital W, small w, then you will get half of the flowers will be violet and half of the flowers will be white. So you will get 1:1 ratio here. If you get 1:1 ratio, then you can tell the genotype is heterozygous. Okay, so by test cross, you can determine the genotype of the dominant parent. Okay, now this is the question based on these topics. Okay, what is the question? In a monohybrid cross between heterozygous parent with a genotype capital A, small a, what is expected phenotypic ratio of the offspring? So you have to tell the phenotypic ratio. So expected phenotypic ratio of the offerings will be, so what is given first of all in the question? Here the heterozygous parents genotype is given to you and it is being said that they are being crossed with each other. This is kind of selfing of the F1 generation. So what are the gametes produced by each parent? They will be capital A and small a. Now you can make a Punnett square. So you can write the gametes on one side and you can make all the possible genotypes. Now you can see from this Punnett square, we can see that here you get the capital A, capital. This is homozygous dominant. So this is 1/4. You will get capital A and small a also. You are getting it 2/4, or you can write this as 1/2. And you are getting small a also, which is homozygous recessive. And you get this in 1/4 ratio. So this is 1:2:1 ratio. So genotypic ratio is 1:1. And here the phenotypic ratio is also 1:2. You can see the phenotypic ratio here is 3:1. So a phenotypic ratio, a dominant phenotype will consist of capital A and capital A and small a. So these are 3/4 or 75%, and recessive phenotype will be small a. This is 1/4 or 25%. So phenotypic ratio here will be 3:1 and genotypic ratio will be 1:2:1. Okay, so 3:1 dominant to recessive. So these are some key notes that will help you memorize these concepts. So these are very important here. Mendel carried out his work from up to 7 years. Okay, this is the time period. So he was the first person. Okay, he pioneered the use of statistical and mathematical analysis in biology. He was working on garden pea. He took large sample size, 2 breeding lines, 14 contrasting varieties, and he did artificial and cross pollinations. Okay, he established inheritance law. Okay, he confirmed the result in successive generations. He is the foundation. He laid the foundation of modern genetics and he proved the inheritance patterns. Okay, now his work was very systematic. He used statistical analysis. His results were reproducible and he gave universal inheritance laws. Now he gave law of dominance. Okay, what is law of dominance? One trait can mask the expression of another trait for same characteristic. A dominant allele masks the recessive allele. Okay. Now, dominant character is observed in the F1 generation, which is heterozygous. Example is purple flower is dominant over the white flower. Then he also gave law of segregation. What is law of segregation? Allele pairs separate during gamete formation. And each gamete receives one allele each pair. This segregation is random and equal, and pure gametes are formed. There is no mixing of the alleles. Each gamete will receive only one allele. And the phenotypic ratio 3:1, this can be explained using law of, this is very important here, question can come from here. So question can come, the phenotypic ratio 3:1 can be explained by which law? So you will write law of segregation. Then monohybrid because it's a cross between parents differing in one trait. Then F1 generation shows dominant trait, F2 generation shows traits in 3:1. So in F2 generation, recessive trait reappears, and you should know the key terms also. What is alleles? They are alternative form of the gene. Homozygous means same alleles, and heterozygous means presence of different alleles. Okay, this is clear. So now we have discussed the theory. Let's quickly discuss the DPP now. Let's discuss this first DPP. Which of the following is not a reason for Mendel's success in formulating the laws of inheritance? So you have to tell which is not a reason which led to the development of laws of inheritance by Mendel. What are the options? Use of true breeding P lines. Second, application of mathematical logic. Third is large sample size in experiment. And fourth is study of intermediate traits. What will be the correct answer here? The answer will be study of intermediate traits. So Mendel did not study intermediate trait. Mendel was studying the contrasting traits. Okay, option number D will be the correct answer here. Then in Mendel's experiment, why were true breeding pea plant variety essential? Why he selected true breeding pea plant varieties? Option, they ensured hybrid vigor, they maintained stable trait inheritance across generation. Option number C, they allowed cross-pollination with two species. And option number D, they increased genetic diversity. Option number B will be the correct answer here. They maintain stable trait inheritance and across the generation, means the same traits were inherited in the next generation as well. The traits were maintained in the next generation. Next question, which pair of contrasting traits in pea plant were not studied by the Mendel? So Mendel studied 7 pair of contrasting character. He chose 7 pair of contrasting character. Which of the trait was not studied by Mendel? Green and yellow pods, round and wrinkled seed, terminal and axial flowers, and purple and white flowers. Okay, so here you can see this question is little bit dicey, you can say, because here the correct answer is purple and white flowers. So in your NCERT textbook, it is written violet and white flowers. That's why the correct answer here is purple and white flowers. Mendel studied all of these characters, okay? In your book, it is written violet and white flowers. That's why the correct answer is here. This is purple and white flowers here. Next, Mendel laws were formulated based on experiments involving what? Cross-pollination of pea plant with intermediate traits, statistical analysis of small sample size, artificial pollination of true breeding pea plants, and observation of natural pollination in garden peas. What will be the correct answer here? So here the correct answer is option number C, artificial pollination of true breeding pea lines. This will be the correct answer here. So Mendel conducted artificial cross-pollination in true breeding pea lines. Now, this one question, what critical approach did Mendel employ that distinguished his work from earlier studies on inheritance? So, what was the innovation done by Mendel? Okay, why his work is considered very significant and robust? Option, focus on quantitative traits. Next, he used mathematical and statistical analysis. Next, studying animal models alongside plants. And last one is emphasizing environmental influences. What would be the correct answer here? Okay, the correct answer here will be option number B, using mathematical and statistical analysis. So Mendel pioneered the use of mathematical and statistical analysis in his work. Okay, so with this, we have discussed the DPP also. Okay, so this is the end of this session now. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.